Okay, good morning, good afternoon, and good evening to everyone joining us from around the world. My name is Sabrina Welsh, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for this webinar. Let's start today with an update on the status of vaccinations around the world. More than 316 million vaccine doses have been administered worldwide, which equals to 4.1 doses for every 100 people. There's already a stark gap between vaccination programs in different countries, with many yet to report a single dose. 10 different vaccines are being administered around the world, according to Our World in Data. And the Pfizer-BioNTech vaccine, which was found to be 95% effective at reducing coronavirus infections, is currently being used in 71 countries. Several more countries have approved vaccines, but have yet to begin administrations. Additional vaccine candidates with different platforms are in the pipeline, and while many vaccines are given as injections, some vaccines can be taken as a pill. Oral vaccines have been approved for diseases including polio, cholera, and typhoid fever. And today we'll hear about Vaxart's work developing an oral vaccine for COVID-19. Just a quick note before we begin. The information presented today includes some pre-published data that is currently under peer review. At the request of the author, we ask that you not reproduce or disseminate the data presented. This session will be recorded and available on our website and our social media channels for you to review. Today's speaker is Dr. Sean Tucker. Dr. Tucker is the Chief Scientific Officer and Vice President for Research at Vaxart Incorporated. He is responsible for research to advance vaccine delivery methods and characterize immune responses to antigens. Dr. Tucker is focused primarily on mucosal immunology and has published several papers on vaccines and immunology. He completed his doctoral degree in immunology at the University of Washington and holds additional degrees in chemical and biochemical engineering from the University of Washington and University of California, Berkeley. In today's lab meeting, Dr. Tucker will talk about Vaxart's efforts to develop an oral vaccine for COVID-19. During the presentation, please send me your questions using the Q&A function in Zoom. I'll ask our speaker a broad selection of your questions after he's finished the presentation. We'll have about 25 minutes for discussion, so I hope you'll take the opportunity to ask your questions about the data presented today. It is now my pleasure to introduce Dr. Sean Tucker, Dr. Sean Tucker from Vaxart. Dr. Tucker, good morning.
Good morning, good afternoon, everyone. I'm going to share my screen now. And thanks for paying attention. So again, thank you so much for the invitation to speak. I'm going to tell you a little bit about our vaccine work. Before I begin, just a reminder, we are a public company, so this is a scientific presentation, and everything here is essentially forward-looking and about things that we may do, and it's not meant to be for investor guidance. Let me tell you about our approach. I mean, essentially what we wanted to do is really work on human immunology or human vaccinations and do something that would be easy from the standpoint of world basically getting enough herd immunity out there. And what we came up was basically the easiest thing we could do or think of for delivering vaccines to people was to put it into enterically coated tablets. And this is what it looks like, and this is in a little blister pack. And obviously, when you put something that's in a container like this, we believe it's room temperature stable for years. We've known it actually can do really well at even elevated temperatures for several weeks. And we think this is a really great way to basically give vaccines out to the world. And it's certainly the most simple, elegant way you could do for adults. Now, to make it work in this fashion took a lot of work and basically on the biology side of things. I think you're aware that there's definitely a lot of oral vaccines out there in terms of the market. There's several that have been approved. And there's a couple problems with those things. Well, first off, they all work by replicating attenuated pathogens. So basically you use that pathogen to elicit a very strong immune response. And when there's certainly some issues, if you want to go very quickly from one antigen to the next, it's really difficult to do that if you're making a very strong anti-vector response. And so what we came up with sort of is an approach, we think of it as sort of stealth technology, that we're delivering something that's sort of, the immune system doesn't really recognize as foreign. So we found out that if you deliver non-replicating Ad5 to the intestine, you don't really get an immune response against that vector, as well as the antigens you're expressing. And what we found is that if you put a double-stranded RNA adjuvant, or basically TLR3 signal, you could basically get a very specific immune response to whatever antigen you've cassetted in. And this is a very, obviously very great from the standpoint of being able to use this technology again, because you aren't getting sort of the anti-vector response you get from an injected adenovirus or for a replicating pathogen to the intestine. And this allows you to go very quickly from one indication to the next. The other thing, of course, is that, there's a lot of issues with the fact that all these attenuated replicating pathogen orally delivered don't do as well in places with where there's, that are impoverished and the intestinal space isn't really well developed. We think that if you don't need replication, this technology may work a lot better. And certainly there's some things that we're going to test. So before I talk about COVID, I'm just going to talk briefly about some of our experience in other pathogens that are respiratory in nature. And this is probably one of the best illustrations to date, essentially, that this technology works a little differently. A few years ago, we were funded by Uncle Sam, essentially BARDA, to basically compare our vaccine approach in tablets to the comparator, which would be Fluzone IM, which is basically essentially a protein quadrivalent vaccine. And because it'd be pretty obvious that you were taking a tablet versus a shot, of course, we had to mask it with placebo injections or placebo tablets. And again, the key thing from this standpoint was we decided we were going to wait more than 30 days before we challenged everyone, essentially gave everybody that was participating in the study flu by dripping it into the nose. And that was done 90 to 120 days after single immunization. Now, the primary readouts in this study were essentially illness, but the other secondary endpoint is infection. And infection here is defined by how much virus is shed or influenza post-immunization. And I think that's very important to think about as well. First off, in terms of the adverse event profile, I can show you that, obviously Fluzone is an approved product. It's very well tolerated. There's no problem. Our vaccine did just quite well. As there was no real malaise, fatigue, or headaches or fevers, with any of these approaches. The only difference is that if you got Fluzone, your arm hurt a little bit more. But basically, the vaccines were well tolerated, and there wasn't very much difference between our vaccine and placebo. In fact, there was none that we could tell. So this very benign sort of technology. In terms of the primary outcomes in illness, our vaccine did quite well and compared with Fluzone. Statistically, there's no difference, but our vaccine had a 39% reduction in the illness rate compared to 27% with Fluzone. And when we look at infection, and again, it's defined by percent of subjects that are shedding 36 hours post-challenge to essentially remove any pass-through virus, what we found is that our vaccine did quite well. There was a 48% reduction in that infection rate versus 38% for Fluzone. And there's an 80% chance that we outperformed Fluzone. And again, we were doing this study with one of the largest, essentially vaccine studies, challenge studies ever performed. And here we were able to essentially find that there was significance when you're looking at shedding, and shedding meaning, basically how much flu virus is basically being put into the respiratory tract. And we think this may be important because it may be that a mucosal response is actually very important from the standpoint of blocking shedding better than an injected vaccine. And again, one of the other things I wanted to point out is that, the other thing that was very statistically significant is that our vaccine didn't do as well in terms of making serum neutralizing antibody responses. In fact, we had about a 10th or a 6th the amount that Fluzone did. And yet we seem to do better from the standpoint of illness, or it seemed that we were doing better with illness, and certainly we're doing better from the standpoint of infection. And in fact, it really correlated our vaccine to whether we were making a mucosal immune response. So when the mathematical modelers looked at all the different parameters we were measuring, what they found with our vaccine was antibody secreting cell IgA class. Those that appear in the peripheral blood 7 days post-immunization correlated the best for protection against shedding for this vaccine, basically making a B cell of IgA class against HA, but not necessarily neutralizing, was an important feature. Whereas for the injected vaccine, it was very clear that HAI was absolutely the most important feature. Basically, when you're injected vaccines, making an antibody response that blocks the interaction between virus and cell was actually the most important for that injected vaccine. And so when we did a logistic fit analysis just to say, sort of roughly correlate what does it take to get to what level of protection, and keep in mind these are order of magnitude estimates, it took about 100 IgA ASCs per million to basically translate to a 90% protection rate. If you do the same sort of thing with Fluzone and talk about HAI, you'd need titers in the 2,700 in the serum to basically equal the same 90% protection rate. So again, what this says is that a little IgA B cells goes a long way on a mucosal vaccine, whereas against this respiratory pathogen, whereas you need a very, very strong serum response to get the same level of protection again, against this respiratory pathogen. The other thing I wanted to notice is that there were some differences between the B cells that were induced post-immunization between Fluzone and Vaxart vaccine. And certainly both vaccines were able to produce a plasmablast, and both vaccines induces in very strong numbers, including IgA responses in the Fluzone vaccine. But what was very clear is that our B cells that were activated had a high upregulation of alpha-4 beta-7, compared to Fluzone. Again, putting the mucosal homing receptor on those B cells correlated very well to protection against this respiratory pathogen for our vaccine. So again, all this data suggests that an oral vaccine can work, and it may work differently than we think of in terms of the vaccine, in terms of immune responses that may be protective. So as I shift back into COVID, obviously, when we started this program back in January of 2020, the problem was a lot simpler than it was today, where there's definitely a lot of strains that have shown up and some strains that are very scary in nature. I think, as let's face it, the vaccine developers have made some incredibly and fast, countermeasures to address this. I mean, it's been incredible that they've been able to get through phase 3 in less than a year, and there's definitely some efficacy, particularly, of course, when the strains match the circulating strain of interest. Obviously, Moderna, Pfizer, AstraZeneca, Janssen, they've all show efficacy, and so does Novavax. But one of the key things is that there has been a drop-off in efficacy noted when the strain doesn't match the circulating strain. For example, some of the vaccines don't do as well against the South African strain, where there's mutations in the receptor binding domain in terms of efficacy. Now, in the US, we're doing actually pretty well. I think we're up to 2 million vaccinations a day. When I made the slide, it was only about 1.2, but that still means that it takes a long time to put shots in the arm. And again, the key thing from the standpoint of responding to a pandemic is not making vaccine, it's basically getting to herd immunity. And if the rate limiting step is how fast you can put shots in your arm, that's going to be your rate limiting step. This thing that scared me the most is basically a lot of these mutations are showing up without actually vaccine selective pressure. There's still not that many people that have been vaccinated. And now there's already escape mutants showing up when those that basically have mutations in the receptor binding domain. As a result, a lot of the vaccine manufacturers, including us, are thinking, hey, do we need to basically update our vaccines to match the strains? And again, this is starting to get into a point where I kind of thought of this as sort of a hamster wheel, where strain emerges, you then make your countermeasure, then you start getting everything out and distributed. And the hope is that basically you can get fast enough on this hamster wheel that you can keep up with all the mutations that are showing up. And again, as we build up manufacturing capabilities and the ability to respond very quickly, that's great. But the rate limiting step is going to still be how many people can you get immunized in a day or in a week or months, because that's going to be the rate limiting step. And you hope that you'll be able to keep up with this approach. And obviously in the US, having something that you can actually give out every 6 months or to update the strain may be doable, but I don't think this is actually how it's going to work in the world. I just can't see people taking a vaccine that's by shot every 6 months. So what we did is we took a different approach. Of course, we give out tablets instead of shots. We think that you can go 20 to 30 times faster in terms of getting it out to the people by handing it out. And in fact, at some point, you may be able to send this by mail to your home and get instantaneous, essentially, vaccination. And we think that's certainly a huge advantage from the standpoint of getting to the end goal, which is promoting herd immunity in large numbers of people. The other thing that we did is we basically took something, that we took an approach where we tried to drive things toward conserved antigens. And so we included not just the S protein in our first candidate through, we also included the N protein. And of course, there's a lot of mutations in the South African strain when it talks to the S protein, but there's only one mutation in the N protein. And we thought that if we could drive a T cell response against something conserved, as well as, providing the S, we'd be able to produce something essentially that may be longer lasting and much more cross protective, and be able to address things in the world that pop up. So again, as I mentioned before, our first candidate through, we included both the S and the N. We have made other candidates that have just the S protein. We thought that there was some advantage. And back in March of last year, one of the things that I was worried about was basically I thought the S had the potential to mutate, and I wanted to make sure that we had something very conserved for a T cell target. With our approach, we knew that using recombinant adenovirus with the TLR3 adjuvant, we would really drive things toward a Th1 response. So we weren't really worried about making too much of a Th2. But again, it's always good to see the data. Before I talk about clinical data, let me tell you a bit of our experience about hamsters. We did a hamster challenge study, sort of back in a few months ago. And obviously what we wanted to do is deliver our vaccine orally, and we can't give hamsters pills, but we gave them a liquid gavage. And we also wanted to compare it to intranasal delivery because we weren't 100% sure that you could actually gavage, a hamster. It can be kind of tricky to sometimes to use rodents in liquid formulations. But it seemed like in this experiment, we were able to reduce antibody responses in the sera, and they did quite well, whether oral intranasal delivery. After challenge, the no vaccine groups also came up in terms of the titer. If you look at the Syrian hamster in body weight, you can see that whether intranasal or oral delivery of vaccine did quite well. The hamsters actually gained weight after the 5 days that we monitored and pulsed the challenge, whereas the untreated animals certainly lost a lot of weight. If you look at terms of the titers in the lungs, you can see that there's a big difference between intranasal oral delivery compared to the no vaccine group. There's about a 4 to 5 log reduction in those titers in the lungs 5 days post-immunization. And again, I think I want to stress that the oral did just as well as intranasal in terms of these protections. And lastly, just wanted to talk about the hamsters. If you use 2 oral doses, there was really no inflammation. There's a big difference between using the mock immunization where there's lots of inflammation in the lungs, and intranasal did quite well as well. There's a little bit of inflammation, but certainly nothing like, the mock immunization. So essentially this vaccine worked, and it worked in a hamster model, and it protected quite well. Now I'm going to switch into our clinical studies, and just keep in mind when we did this study, we have basically had the same sort of issues that everybody else, that there's supply issues for, making our vaccines. And we were only able to do a very small study from the standpoint of doses. And so this is a single dose study, what we consider at the low and medium doses. And they were all single doses, except for one small cohort in the low dose that was boosted. In this study, we did meet our primary and secondary endpoints. The vaccine seems to be well tolerated, and certainly it seems to be very immunogenic. With that, let me dive into the data. So first off, solicited symptoms post-vaccination. You can see that there's really not much, to be concerned about. There was no serious adverse events, no adverse events of special interest. There's more randomness in terms of the unsolicited AEs, and in terms of the solicited AEs 7 days post-immunization. There's really nothing that's different, or there's nothing that really concerning. Obviously it's well tolerated. There was possibly a slight increase in the abdominal sort of symptoms, but certainly nothing statistically significant. And again, one of the key things about this vaccine, we don't seem to make the same amount of headaches or cause malaise as it's been reported for other vaccines. So we think that this vaccine is well tolerated and can go on. When we look at the T cell responses, I'm going to focus on the T cell responses for a little bit. Obviously, by including the nucleoprotein, we certainly make, or nucleocapsid, we certainly make N-specific responses. Putting in the spike as well, obviously, we make S-specific responses. And it seems to be very driven toward Th1. We're making gamma, TNF, and CD107A. If you look at the gamma in responses, what was very striking to us is just how strong the responses are. Certainly there's a few subjects with a high background, but we're getting some subjects that basically have somewhere in the order of 4% gamma interferon above background, or 1.5% on average. And this seems pretty substantial. And it's very, very S-specific in terms of the response. If you actually take those same PBMCs day 1 and day 8 and use sort of a control peptide against, conserved CMV Epstein-Barr flu, you'd only get about 0.5, 0.8% in pre, and it's the same pre and post. So again, we're basically increasing this S-specific responses, and we're making some that are very, very strong in terms of the percentage of CD8 gamma that make either gamma or TNF. This is pretty robust. This is like the best we've seen in terms of a T cell, CD8 T cell response. So again, from the standpoint of COVID, why could this be important? First off, T cells can be cross-protected and long-lasting. Certainly N protein-specific responses to SARS-CoV-1 have lasted 17 years after infection and can cross-react to SARS-CoV-2. We know that T cell response can reduce the severity and length of infection. And then I just heard this, a few weeks back that people that have no B cells and no antibody, they actually do okay. They have a mild course of infection and don't die of COVID. That's actually quite significant if you think about it. We know that mucosal T cells are capable of blocking shedding and transmission. We know the mucosal responses do that as well, and something that we're very seriously exploring right now. And there's definitely lots of data out there in the literature that suggests that a T cell response could be very important. And obviously, because you're spreading the whole response over the whole S protein, they can be much more cross-reactive than when you're looking at antibodies, where it's very important. And certainly from the injected standpoint, you know, the business end is basically driving those antibody responses against the receptor binding domain. So again, we think that building a cross-reactive, longer-lasting vaccine such as this could be very important. Now, let me dig into the antibody responses. And again, you know, we were very surprised in how strong the T cell responses are. We're also a little bit surprised, you know, about the antibody responses. We certainly make increase in plasmablasts, you know, in the study. And we certainly see a dose-dependent response in terms of making IgA-ASCs, but also basically increasing the base cells that have either IgA and alpha-4 beta-7. And again, from our prior work and, you know, a different, you know, respiratory pathogen, this was a very important correlate. When we actually looked in the serum, we actually saw no neutralizing antibody responses whatsoever, but we did see that there were some increases in the S-specific responses, receptor binding domain specific response, or N-specific responses in a mucosal compartment or in the serum, but mostly in the nasal response. We saw very much strong increases in some subjects, and we think this is, again, pretty important. So just as a summary, and again, I wanted to try to get through this very quickly so people could ask questions, but you know, we know that the vaccine, you know, our first candidate through for COVID-2, we met the primary and secondary endpoints. We know it's well tolerated. Obviously, tablets are very easy to distribute. It was highly immunogenic from the T cell side of things. On the B cell side of things, you know, certainly we didn't make the serum responses that we expected, but we did see a dose-responsive responses, and we think that with subsequent studies, we're going to look at formulation and dosing to sort of really boost those mucosal responses, because we think could be important. Obviously, when we started this is a little different in the world right now. The US is going to be immunized, I said 6 months, but you know, it may be on the order of more like 3 at this point. And so we need to basically make sure that the vaccine, you know, will be able to boost people that have been priorly immunized in this country, as well as build a product that could be used around the world. You know, lastly, one of the things we're doing is we're evaluating cross-reactivity from this, you know, from the serum mucosal, you know, samples as well as the T cells. And we want to make sure that we're building new variants as well that can address potentially escape mutants. So we certainly have a lot of things that we're very interested in and how to move this forward. With that, let me just thank the people that basically helped in this study, you know, all the team at Vaxart, Stanford University, and our collaborators at Cork and Vizimaderi and Loveless. And again, I tried to get through this so that we'd have time for questions. So with that, I'll stop the presentation. Thank you.
Dr. Tucker, thank you so much. It's so interesting to see this new data and to see how it all came together is fascinating, especially in the context, as you said, of this really rapidly emerging development field. So thank you for sharing that with us. We've got a lot of questions, so I'm going to dive right in.
Great.
Okay. I think you may have touched on this. When can we expect final phase 1 results, and are you planning for a challenge study?
So the short answer is, yeah, we are expecting to basically sort of write up a lot of the phase 1 results. We're sort of following up on some of these things, you know, doing some comparative studies in T cells, looking at cross-reactivity. You know, we certainly would like to get this all put together. And, you know, I don't think we provided, you know, again, guidance, but, you know, it's on the order of months for us to sort of finish up and put a paper together. We've tried as a company to do a good job of publication because, and it's not easy to do as a company, but certainly we've historically been tried to be very good about it. So our plan is to get this forward. And you said something about a challenge study. Yes, I think that would be a great idea. And, you know, certainly from the standpoint of our vaccine, you know, the challenge study could be very useful way to look at efficacy. Our guidance that we've told people is that our plan is to do a phase 2 study and dose ranging study. And if that looks good, to take that forward and do essentially a field efficacy study, either phase 2B or phase 3, depending on the size. But the challenge study is certainly entertaining it's interesting to us. I don't know if it's ready for prime time, but I'm not going to say that we wouldn't do it.
Good. It would be really interesting for sure.
Yeah.
Okay, I'm going to go through some of more of the vaccine trial design questions first. So I think they're all kind of related to each other. Will you be looking at mix and match strategies where your vaccine might be the first or second dose in a series with another injectable vaccine?
Yeah, I think there's definitely merit to basically having our vaccine as using as a, certainly at this point, the injected vaccines are ahead of us. So I think we have to really address and actually ask the questions, how does our vaccine approach work in terms of a boosting vaccine? And certainly we think that it's very important, in countries that are well ahead on the, how should I say, in terms of rolling out the immunizations, and the US has done quite well. And that the reality is, the world probably is going to have a first dose vaccine, in the next 3 years, I would guess. So yeah, it's very important. And I think we'd love to be able to see how it works. And because the immune profile that we get is so different than what the injected vaccine, I think this might be, it'd be very interesting, question to ask.
Definitely. Okay, so a good question from a few different participants. Can we use this both as a vaccine and a therapeutic? Would you consider your pill, prophylaxis or as a booster only?
Yeah, I don't think that we've developed the technology from the standpoint of a therapeutic. I guess the question, of course, is, how fast could your T cell responses come up, once you've been exposed? And I don't know the answer. I mean, I understand, obviously COVID-2, it seems to be a slower growing virus than flu. So I'm not going to say it can't be done. I just don't have any data. Obviously, we think that this technology is very good from the standpoint of getting vaccines out to the people as a prophylactic, and we think that that's the way that the company has sort of positioned it for the short term.
Great. Okay. Well, let's see. Lots of questions. Thank you guys for being so engaged. This is really, really helpful and nice to see. Okay, let's look at data questions now. As one of the secondary endpoints is the measurement of serum neutralizing antibody, how is this endpoint being met, and is there no serum neutralizing antibody found? I know you commented on the news before.
Yeah, I mean, obviously, one of our secondary endpoint is immunogenicity, and there's certainly several different ways of measuring it. We did really well on the T cell side of things, and we didn't, do as well on neutralizing antibodies. We think that part of our approach, that how we designed this vaccine, may have driven those T cell responses, and maybe at the expense of the serum antibodies. So that's definitely a question. Or it's all about dose ranging. So, when we do higher doses and boosting, we may get a different profile. There may be some sort of threshold event. We're just not sure at this point. One of the big limitations in this study is that we only had enough vaccine to do a lot of single dose and very lower dose. And so, but those are things we can explore in the future. We, certainly solve some of the supply issues, and we were moving it forward.
Great. So on the approach, with antibodies, durability and protection can be measured. Is this possible with your approach? Can you look at long-term durability and protection?
Yeah, that's a good question. I think it's a little more challenging from the standpoint of, from the T cell side of things, because obviously most of the time what you do is take out the peripheral blood, you look for, responses, and then you can measure it. I mean, that's doable. There may be ways of basically sort of looking at a T cell profile that may be important. I don't have any data to say how we might do it. I just think in theory that there's ways to do it that are a little bit better than pulling out PBMCs, doing your pulsing with your peptide libraries, and measuring gamma interferon. So.
Okay. All right, let's move into some mucosal questions. Does the oral vaccine induce effector resonant memory T cells in the respiratory tract, or are they somehow recruited from the intestine after infection?
Yeah, that's a good question. I mean, I don't have the answer from the standpoint of humans, because I can't pull tissues out of the nasal, people don't want. It's funny. I ask the chief medical officer all the time, can we pull some tissues, and just against healthy people?
Noses? Yeah.
Yeah. And for some reason, no one wants to volunteer, people are very reluctant. In animals, what we have done is we have been able to pull out, some of the tissues for the respiratory tissues and look for, in this studies, like for example, in the cotton rat study with RSV, we were able to pull out tissues, tissue resident B cells and find antibody secreting cells. We didn't really focus on the T cells for RSV because we thought antibody was more important, but certainly you could possibly do that in animals. It's just, there's just limitations what they'll let you do in humans or people volunteer for, which is fine. I think we're okay with that. So.
Well, measuring that, that's obviously challenging. So there's a question now about the GI involvement. We know there's GI involvement in SARS-CoV-2 infection. Do you think this vaccine will protect at the GI level? Is it protecting in the gut?
Obviously, yeah. Our expectation is that if you can get the mucosal responses up, you're going to be able to make a mucosal response in the intestinal and, or in the intestine, and that should be able to basically sort of suppress or, get rid of the SARS-CoV-2. We do know that people that there are indications in the literature that people basically shedding for long periods of time in their stool, most people believe that, of course, the respiratory approach is much more likely to be the way it's transmitted. But I think with other coronavirus, there's documentation that people have been able to spread it by the fecal oral route. And, again, from the standpoint of one of the things, we'd like to do, of course, is, with this sort of mucosal response is, can you use this to sort of block shedding and transmission? And if you can do it both in the respiratory as well as the intestinal tract, that would be great. I mean, I think we'd all like to have a vaccine that can block transmission as well as, in protecting the subject.
Mm-hmm. Definitely. So I sort of had a question on, looking at oral vaccines over the course of the last few years, it seems like there's so many practical applications of these in terms of, does it require refrigeration? First, that's a good question to start with.
Yeah, I mean, our approach basically, adenovirus itself is very stable, and when you dry it, it's incredibly stable. So you can put everything in things, and we have data that goes to 2 years that, shows there's really no loss of activity, using other indications, but using the same sort of technology. We know that we can take elevated temperatures, and in terms of stability, this is a lot better than, most of the injected vaccines. So
yeah. So the potential applications for low and middle income countries where it's harder to get refrigerated injectable vaccines could be fantastic. So LMICs could really benefit from this. So I have a question about the implications for people with comorbidities, specifically, infestation with gut parasites. There are a lot of helminth infections in East and Southern Africa and all over the world, really. So would a helminth infection or a comorbidity with different gut parasites, would that affect immunogenicity and how someone responds to this type of oral vaccine?
Well, it's certainly an open question, and something that, we sort of like, what we think is that people that are in impoverished region where there's a lot of infections, the gut intestinal biology doesn't make the same villi. There's a lot of issues. And certainly that we know that, if you need a vaccine that is replicating, like all the oral vaccines to date have been replicating attenuated pathogens, we know there's some issues. We think that there may be some pluses and minuses to our approach. On the plus side, if your intestinal villi is not well developed and the mucosal layer strip, it might be easier for our tablet to get in because we don't need to replicate all the interferons that are being secreted that basically may block replication, may not be as important. But certainly, we don't know for sure, what will happen. Our expectation is that we'll have to do some dose ranging studies because it could work better than it could in the not so impoverished regions, or it could work worse. And it's certainly something we're very interested in exploring. I mean, obviously, you pointed out that, from a tablet standpoint, getting out there is certainly, it would make things a lot easier. And even from the standpoint of just distribution, I've had discussions with sort of, large government entities, and they always say, what's your vaccine cost to make? And I say, well, the cost is not necessarily just what the vaccine takes to make, it's how long it takes. It's all the costs associated with, cold chain distribution, immunization. Absolutely. And even in countries that are, a little less wealthy than the US, there's still a cost to that. And if you can basically, get the overall solution so that it's still competitive, then I think it's still a wonderful way of going about it. So,
especially if you can reach people who wouldn't be able to benefit from a traditional injectable vaccine. There's a huge impact potentially there. Are you doing studies in LMICs and potentially with people who have those types of comorbidities?
So the short answer is that we are trying to open up regulatory pathways in countries that are not, as wealthy. We have, would love to work with, entities that have a lot of resources and expertise in doing these things. Obviously, we're still a medium-sized biotech company, so we can't do it all, but we're very open to working, especially on the COVID side of things, because we do think that there's a huge benefit to having a long-lasting cross-protective solution that doesn't require, we can contemplate having a shot every 6 months for COVID in the US, and it's not out of the question, but most places in the world, that's never going to work. And so we'd love to be able to have discussions about how to get this out there because, again, we think it's a different solution, but it's still a solution.
Absolutely. Okay, whew, tons of questions. Thanks, guys. Okay, more mucosal related questions. Are you measuring antibody and nasal washes of vaccine recipients? And someone noted you could do, where was that note? I think nasal lavage or, yeah, human lung lavage washes for studies in vaccinees. Is that possible? And are you looking into that?
Yeah, so we have not done the full deep lung lavages. I know that's possible, but you know, people tend not to want to volunteer for those studies, so we've avoided it. Historically, we've done nasal washes, and we published a paper on flu, and we were able to see pretty good responses. The problem with doing the nasal washes is the material that comes out can be pretty dilute, and you got to basically account for the fact that there's a yield. So you need to measure total IgA as well as your specific IgA, and then sort of normalize it to basically make sure. What we've done recently is use nasal swabs, and that's actually done pretty well from the standpoint of being able to look at responses. In this study, we didn't go through details, but the nasal swabs were getting a lot of IgA responses in subjects that we can see that are increasing, again, especially against the receptor binding domain, but also the S protein in general. So we are seeing those responses. And the next study, what we would like to do is, of course, at higher doses, we'd like to really see, if we can bring those up a little higher. Because again, as you pointed out, we'd love to be able to have a mucosal response, not just in the intestine, but also in the nose. And we think we can do that, so.
Great, so this is an interesting question. There have been suggestions and some evidence that mild enterovirus inoculations in the gut can induce IFN and reduce respiratory mortality. Do you see your formulations used in this way?
Yeah, I mean, that's a good question. And the short answer is it's theoretically possible, but of course, if you're going to talk about doing that sort of approach, that's a very short-term thing. So you'd have to be giving, if you're trying to do it prophylactically, you'd have to be giving tablets all the time, and that may not be practical in most places of the world. But but in theory, if you think that you've been exposed to, just hypothetically, maybe if you're exposed to some virus and you think that there's an advantage of making these sort of interferon responses, you could start taking the tablets prophylactically and see if there's a cross-reactive response. I mean, I look at things more on the adaptive side of things, and so I haven't thought this through, but I mean, maybe there is a merit to doing it, so.
Okay, sure. Okay, a couple of questions now. I think you commented briefly, but can you expand on the shelf life? Is it months? Is it years? And in terms of distribution, how does that work?
I'll give you a scientific sort of approach, not a regulatory thing. So obviously, we know that this vaccine platform can last several years at room temperature 25 degrees. We know that is true. It'll stay in spec, or at least we have data to show it. Obviously, the key thing from the regulatory standpoint is you have to prove it to the regulators that your vaccine has that kind of shelf life before they'll allow you to put it on your label. So the scientific question is, it's years, at room temperature. You can take, we know that it can tolerate some higher level temperatures for short periods of time. But, from the regulatory standpoint, you have to basically prove to regulators that it has that stability. And then, in terms of distribution, well, the way I think about it is, how long does it take you to go to get your, when you go wait in line for a COVID vaccine, you get in your car, you drive in the little thing, they give you a shot, then you got to wait. Well, I mean, from the standpoint of, distribution of a tablet, you could do sort of the same thing. You could just hand out, your little blister pack with your little tablet, and do the same thing. And in the long term, if you basically get to the point where you feel like the safety profile is such that you don't have to worry about it, you could send it by mail. I mean, and you can instantaneously immunize, a huge populations. Or, if you're worried about the postal service being thwarted, maybe you could send it by Amazon drone or something. But I mean, there's lots of different.
Someone commented on it earlier, like you could even just drone to different places and drop it off. Essentially,
it could all be done, but of course you want to do this in a very safe and regulated fashion. So you'd probably start out by just handing it out at the same sort of clinics that you would basically have shots. And our estimation is you can go 25 to 30 times faster in terms of handing out pills that you can give a shot. Just that's our estimate. But obviously, at some point, you can get to the point where you just send it. I mean, there's no reason that we think that it can't be done. They send heart medicine through the mail. Why can't you send a vaccine?
Yeah, sure. Well, an old colleague once said, if you can, you can get a refrigerated Diet Coke or Coca-Cola in the middle of Sub-Saharan Africa, we should be able to get a vaccine there too. So I always like to think about that and keep that in mind. So a few of our participants talking about our discussions with LMICs and access, how do you get the Vaxart pills? I'm sure there are regulatory issues involved, but is there a contact at Vaxart for people who want to engage with you to learn more about either setting up a study or purchasing for a region? How does that work?
So we are, if there are places in the world where someone wants to basically go and, work with us in terms of moving it across the finish line to getting a phase 3 study done, or to basically provide buy vaccines. I mean, obviously those are all business contacts. We're completely open to it. I mean, we think, we'd love to be part of the world community, so we'll do it. I mean, for me, if someone sends me a note and says, hey, we'd love to be able to participate, and here's, the resources we have, you can send me a note, I'll pass it on to business people, they'll get it, they'll make something happen. So
great. All right. So you can email Sean, everybody.
You can email me and I'll pass it on to the right people. But yeah, we're, like I said, we are very open to, participating, in all around the world, not just in the US.
Super. And I don't know, can you comment on costs of the vaccines versus other traditional injectable vaccines? I mean, we talked a little bit, it's sort of hard to estimate because it also includes cold chain and all that jazz, but.
Well, I think that they don't let the scientists basically list costs, because we're always going to say, yeah, we'll just give it away for free. So, but I would just say that in the overall grand scheme of things, it's going to be cost effective from the standpoint and in line with all the rest of the COVID vaccines in terms of when you look at the whole, manufacturing as well as distribution. We think it can be very competitive with pretty much the technologies that are out there.
Yeah, great. Okay, so in terms of delivery, we've talked a lot about pills and tablets. Are there any plans to deliver in liquid form for children or other forms? Can this be constituted or reconfigured for children?
We're working on formulations now that basically, allow it enterically coated microparticles. I mean, obviously, one of the things that we like the idea is something where you have, I think, Pathway had developed to something where they had basically a packet where there was a solid dose form, in sort of granules, and there was some sort of a liquid buffer. You break the packet, you mix it around, you tear its thing, and then you can give it to kids. So we think that would be doable with this technology. And obviously we are working on these things, but it's not ready for prime time yet. With COVID, I think that, obviously the adults are the more risky population. So we're focusing first on adults, but certainly we think that our technology would be important for kids. And our norovirus program, we think it would be very important for kids. So that's essentially a must do, from the standpoint of getting this out there.
Absolutely. Could you comment a little bit more about the norovirus efforts? Because a couple of our participants were curious, there were a few slides earlier. Could you say a bit more about that effort, please?
Sure. So we have a norovirus program. We are working on a bivalent approach where we're targeting a G2.4 as well as a G1 strain. We did a bivalent phase 1 study. There was no interference between it. We obviously with COVID, everybody put everything on hold except for COVID. So we put our norovirus program on hold as well. We just announced recently that we've restarted it. We're doing a boosting study. We're doing an elderly study. And our plan is to potentially do something on the order of a phase 2 challenge study, later. Right now, of course, with COVID, it's still a little difficult to get a lot of people to volunteer for studies potentially, but it's getting better because as the people that are getting the vaccines, they're more willing to go into the clinics and basically participate. So we are starting studies. We have announced we're starting studies, and we're moving it forward. And like I said, we'd like to get to the finish line on this. And with the tablet, of course, I think most people will buy that a mucosal intestinal immune response may be very important for something like norovirus.
Are you aiming then to conduct one of the very large phase 3 trials, like the 30,000 subjects trials, after a phase 2A or 2B is positive?
Yeah, I mean, the regulatory path, we haven't disclosed exactly how we plan on doing it, but certainly a traditional path of doing a large phase 3 with norovirus, that could be tricky from the standpoint of you have to sort of find, a huge number of people get infected with norovirus every year, but it usually comes out in big sporadic outbreaks. So the challenge is basically to figure out, how to do this in a place that you think that there's a very high likelihood that there will be an outbreak and you can get those efficacy signals. Okay,
gotcha. All right, shifting gears a little bit, could you comment more about the variants? That's sort of not a hot topic, but that's what it's on everyone's minds. So could you talk a little bit about Vaxart's plans? Can you pivot to develop, specific for each variant strain?
Yeah.
And do you have a timeline for the S protein vaccine for the South African variant?
Yeah, I mean, the 20,000 foot question, of course, obviously we thought the variants were was going to be an issue. We put the N protein in because we wanted to make a cross-reactive T cell response, and I think there were certainly advantages to that approach. We don't know if the variants that are coming out are going to be limited. Like there's a certain number of them that basically you have to cover to be able to cover all the existing dangerous SARS-CoV-2, or whether that's going to be infinite. And obviously, the more infinite is, the more a cross-protective approach would be beneficial than having something for that. But we certainly are interested in basically ensuring that our vaccine is cross-reactive. We're basically looking at doing some tests to look at that. But we also are building vaccines that are like S-specific and against the South African. We have no guidance about when we'll be ready to test those things, but we certainly are making them, and we will test them preclinically. And if there's merit, we will move them along into the human population as well. And again, obviously, if you can boost with a tablet approach, against something like South African, given the way that the vaccines are working, I think that would be a wonderful solution. So
absolutely, yeah. So thinking more along those lines, could this platform be used to create a universal coronavirus vaccine? Or
there's
flu
definitely
vaccine that's
merit.
Universal?
Yeah, I mean, there's definitely, from our standpoint, we think that we have some really nice data from this T cell side of things, and we think that we'll be able to provide a package that shows that it's very, very cross-reactive with lots of different coronaviruses on the T cell side of things. The other way of approaching it as well is that if you can create a antibody response in the nose that's because IgA and dimeric IgA is able to basically turn a low affinity interaction to a much more potent because of the the polymeric nature, you may be able to build something that's a little more cross-reactive. Our experience with flu suggests that the business end of the injected vaccines is to make an IgG response that's really against that binding of the cell to the virus. Our experience with flu with IgA is that it may not necessarily mean to be functional in terms of a response. You may not have to have an IgA that binds in that region. You may be able to bind at other parts of the receptor and still have efficacy, as well as, so we hoping, again, we got to prove this, that an IgA response might be much more cross-reactive, than an IgG response, just because, and again, this has to do with what our experience is flu, rather than our understanding of coronavirus, because it's still limited at this point.
Yeah, sure. And did you detect antibody responses against other antigens of COVID other than S and N?
In our
a bit
vaccine,
like flu-inducing antibodies against H and N, so it's sort of a similar.
Yeah, well, obviously, when you only put S and N in, we can only, we're getting antibody responses that are increased to those antigens rather than in the mucosa, not to things that are other parts of it. So, I mean, in some ways, that's kind of a good marker too, right? If you wanted to look to see if your vaccine, if the responses are vaccine specific or someone got infected, you could do that. So, but realistically, what we see is increases in S and N in subjects, some subjects, in the mucosa. So.
Gotcha. Okay. Wow, lots of questions. Let me just take a quick look.
Well, I'm glad
you're officially answering the most questions I think we've had in the lab meeting, so I appreciate your time.
I figured that the best way to basically bring this out is basically to answer questions. I mean, obviously we have, some data in COVID, but, I figured that this would be, I wanted to keep the talk short to really make sure that we had time to, talk and,
well, we appreciate it.
Great.
Okay, so questions about manufacturing. You mentioned, and obviously this is a huge issue for the field right now, that there are limitations on the bags and the piping and all the different bioreactors and all that. So your limitations for manufacturing are similar to the fields. What has been done? What is Vaxart doing to proactively kind of encounter this and figure it out and troubleshoot it? And do you have any lessons learned that the fields could benefit from in terms of scaling up and manufacturing capacity?
Yeah, I mean, that's a good question. I mean, obviously on the upstream side of things, we're making adenovirus like a lot of the Johnson vaccine or the AstraZeneca vaccine, and we're going to competing with similar materials. You mentioned the bags is a big issue for the bioreactors.
Yeah.
So yeah, I mean, and that's a big limitation. We're trying to figure out workaround solutions. We're hoping that the U.S. will start, that the Biden administration may be able to sort of, kind of tell manufacturers to go and incentivize them to basically make a lot more of this, so that, our vaccine trials can be, larger and much more robust. the thing that we actually, there was definitely advantage. Well, some of the advantages that we have is because we don't have to fight for sterile vials and needles. That gives us a major advantage from the standpoint of limitations, because for a while there was not enough glass vials of medical grade to be able to fill syringe vials, and there's definitely shortages of certain needles, and like the ones that were retracting and the ones that allow you to get down into the very bottom of the vial. And the good news for us is that that's at least one thing that we don't have to worry about. So, but yeah, I don't have any lessons learned, but except that I think that if the next time there's a pandemic, maybe it's maybe the reality, maybe what governments need to do is to think about how the manufacturing can be structured so that things that are important from in terms of raw materials, as well as things like just like the bags, could be much more efficiently made. So I think that would be advantageous for us, and the whole field is basically to have find enough suppliers that can do these things and try to ramp it up early in the process, so that we're not worried about hey, how am I going to make this next lot of materials? So
yeah, well, so is there still a supply problem for the phase 2-3 trials, or has that been resolved, kind of making the material for it?
I think we found most of the way we found workarounds, or we found materials. So I think we're in better shape to go forward. But I can't say that there won't be shortages in the future, because it kind of depends on the field. I mean, it's also possible that some manufacturers may decide that they're no longer want to be in the COVID race, and all of a sudden the huge amounts of supplies pop up. So these are things that we have to be aware of, and everybody has to be aware of. But it's all kind of behind the scenes, I think, to some degree.
Sure.
So
yeah. Okay. Well, these are interesting questions. So when we're thinking of an oral tablet, could you potentially combine diseases? So could you make a combination norovirus and COVID tablet together and have it delivered together and stimulate a response to both?
Well, it's interesting because we've had these debates internally, because from a scientific standpoint, you could put a lot of things on one tablet because the active ingredient, is, 0.1% or less. So there's definitely a way to do that. Now, from our standpoint, we're kind of going the other way. We're thinking, well, no one's going to take 3 syringes, if you're, I mean, you can force your kids to have, 3 shots, right, during their immunization campaign, but most adults aren't going to want to take 3 shots to get your flu vaccine, your coronavirus, or whatever else. That just doesn't work. So there's a huge incentive for the needle people to basically put everything on one. We don't think that there's the same issues with the tablets. My 101-year-old grandmother used to take 90 tablets a day or something, and without a problem. So I don't think that having 1 or 2 tablets, or even 4, is going to be it's going to discourage people from taking it. I do think that having 4 needles would be a real problem. So I think that this is a huge, for us, the nice thing about this is that, let's just say the shelf life of our vaccine is more than a year, and you have a, norovirus flu thing, and you may have to do some swapping and changes. We don't have to throw out the whole thing. Once you formulate it, you potentially could be able to, keep your things that are still constant, and then put in the new tablets without having to throw it away. So we see lots of advantages of being tablets and flexible.
Great.
So.
Okay, so I think this is a great question, but I don't know if you can answer it, but I'm going to ask it anyway, because that's what we do in the lab meeting. Okay, what questions have you had from the FDA that are different from the questions they asked to injectable manufacturers?
Yeah, let's think about, let me think about if there's things. I think, obviously, I can't give you any, real, I mean, I'm trying to think what's been public, but I think that, what I could tell you is that our interactions with the FDA have generally been very good from the standpoint of, they ask good questions. They're data driven. They're basically responsive. They want to make sure you're not doing anything that's dangerous. One of the things that was different about our approach, here's something that's pretty much out there. When the adenovirus vaccines, for HIV, came out, there was some guidance from the FDA that they wanted to pre-screen people that had low antibody titers to add 5 for the injected vaccines. And I think that was in a VRC studies. So we've never been forced to basically pre-screen people. And we have data that says that our vaccine isn't hindered by pre-existing immunities in the sera. So I think that's one difference that we've seen, is that of course, it's all data driven, but we've shown that the antivirus responses don't seem to impact our vaccine like they do with the injected vaccines, and we don't seem to get the same sort of anti-vector. So I think the one thing that I could point out is, and this is public, is that we haven't been forced to essentially pre-screen people for catavirus time. So
interesting. I worked on some of those trials for IAVI with the AD35 and AD26, and screening is a part of every protocol. So it's very interesting to hear that's not part of the Vaxart requirements.
That's one thing that I can tell you that, it's been different. But again, as if you're aware, look at the FDA, they ask questions about, and they want to make sure that your approach is, not going to cause any problems. And, and from a company standpoint, they want to make sure you're not cowboys and, and going to do wild things. So, and, that's not been our approach. So, and again, if you provide data to answer the questions, they're all happy. We're all happy. Yeah. So.
Great. All right, so we're getting close to the end of time. I'm going to ask one more kind of general question just to see what your thoughts are on timeline and the pipeline, and where do you see Vaxart a year from now? And in terms of timeline, when are these products coming to the market?
Yeah, I think that, I'll jump to norovirus. We think that obviously we've restarted the program. We believe that we'll be in phase 2, toward the end of this year, early next year. In terms of the COVID, obviously there's a lot of questions that things are changing all the time. The competition there's a lot of people out there. And so, we have to sort of figure out how we're going. What we said we're going to do is we're going to do a dose ranging study with this candidate. And we're going to basically make some decisions about taking it forward, then do an efficacy trial later in the year. We would like to evaluate some S-only constructs preclinically, and if those look very promising, then we would potentially advance those. And again, the key thing also is that we have to basically sort of figure out how these vaccines are going to look in combination with other injected vaccines, because the reality is, we're not first. Most of the U.S. will be immunized by the summer. We're going to have to be able to see how this looks in that sort of, and again, from the standpoint of, we would like to be a late stage company within a few years. So, phase 3 on multiple fronts.
Great. Well, it's really exciting work. Thank you so much for your time and for going through all these questions with us. You officially win, I think, is the most questions answered in the lab meeting. So thank you for your willingness, especially at 7 a.m. California time, to answer all these, and for joining us today. I can't thank you enough.
Well, thank you very much. And I really appreciate you guys inviting me to speak.
My pleasure. And thanks to all of our participants for joining. Your questions really make these meetings so interesting. So thanks for joining. I'm going to grab the screen back and share a couple of slides, and then we will wrap up. Okay. Great. So I invite you to join us 2 weeks from today on the next Global COVID Lab meeting, which is scheduled on March 25th. Our next speaker will be Dr. Penny Moore from the University of Witwatersrand, who will be speaking on the SARS-CoV-2 infection and vaccination in South Africa. I also wanted to highlight an upcoming scientific prize that is presented by the Human Vaccines Project and the Michelson Medical Research Foundation. The Michelson Prize's Next Generation grants are given to support promising young researchers who are applying disruptive concepts and inventive methodology to advance human immunology, vaccine discovery, and immunotherapy research for major global diseases. If you're age 35 and under, you may be eligible to apply. So I encourage everyone to check it out and forward to others who may be interested. I think it's a really great program. We're really proud of it, and the prizes have uncovered some really interesting and impactful science in the past. So we're looking forward to getting this year's prize started soon. The application portal will open on April 1st, and you can find more information on the link I'm showing at the bottom of the screen here. And if you're interested in more research on COVID-19, please sign up for the HVP COVID report. In this newsletter, we present insights from experts all around the globe and highlight the latest scientific articles and data. And finally, please feel free to visit our website and follow us on LinkedIn. We'll upload a recording of the webinar there for you to review. And with that, I'll say thank you again, everyone, for joining. Stay safe, get vaccinated, and we hope you'll join us again on the next Global COVID Lab meeting. Bye, everyone. Have a great day.